Charles Kettering once said, "There is a great difference between knowing a thing and understanding it. You can know a lot and not really understand anything." To share with us the journey of understanding memory in living systems, we have with us Mr. Nikhil Yenugu, PhD scholar, IISER, Kolkata, India. Mr. Nikhil Yenugu is a second year research scholar in the Department of Chemical Sciences at Indian Institute of Science, Education and Research, Kolkata. He obtained his BSMS degree in chemistry with physics minor from IISER Kolkata in 2020. His research interests include quantum chemistry, electronic structure theory, and molecular reaction dynamics. Apart from his academic studies, he has a keen interest in studies at the interface of foundations of science, science of mind, memory, and consciousness. He has been a part of Young Minds Speak. Please put your hands together as we welcome Mr. Nikhil Yenugu.
So today I will just present a timeline of how scientific community is going through understanding memory from last 150 years. So before starting, I would like to dedicate this small presentation to Dr. T.D. Singh. Because of his books only, I got interest in this field. So to begin with, in Aristotelian philosophy, actually Aristotle thought the mind is just a cooling system, and the intelligence is there in heart. So with that ideas, almost around 1500 years people are living with, but this Greek, ancient Greek, he has dissected the eye and saw the connection between the eye and brain, and from there the dissection studies have started, and little by little over the time people have got to know many things about by dissection studies. And out okay, so and even apart from dissection studies, even mental health studies also have started very early from the Persian Empire things. So they are two physicians who were dealing with patients who got injured in the wars, and they were actually trying to relate so many neurological disease with the empirical formalism they were trying. Next. Okay, this is not so. So just to give a very key milestones in neuroscience, because to understand memory, we need to understand the history of neuroscience also. So for that, the electromagnetic nature of nerves has been discovered in 18th century, and later on, and very interesting idea is that specific function of body has been relating to the specific regions of brain. So this idea was already developed in 1800s, and the uh, mapping has been grown, and we got to know neuron and synapses. All these things got introduced to our knowledge. And apart from that, even the at molecular level, concentration gradients of iron also has been introduced, we got to discover. And to study uh, the neuroimaging of brain, now we use three major instrument: EEG, PET, and fMRI for spatial and temporal resolution of our brain, and mapping the events of the brain to the brain spatial parts. Okay, sorry. So to come to our topic, memory. So there are actually three broad class of problems have been involved. So memory, what we understand, mainly three things now. First, we will take the sensory input, and after that, there is a process of organization, and it will store. And after that, whenever we want, we will use that. So this is the general understanding. So uh, in that spirit, there are three areas. This is memory consolidation and nature of memory systems, and nature of memory representations and memory systems. I will, in my presentation, I will cover about only these two topics. This has been already covered by our speakers before. So consolidation has two aspects. One is at the physiological level and one is at the psychological level. So just to introduce that these terms I am going to use. So before using, I would like to introduce amnesia means it's memory loss, and retrograde amnesia means the you will you will not have memory about before events, before the disease has been uh, before the patient has been incurred with the memory loss. And antegrowth amnesia means you will not remember, brain will not have capacity to form new memories. But so to start the journey with, people have actually studied, instead of studying memory, how we remember things, people started studying how we forget things. And they started exploring, like they have started exploring the learning part and the time correlation, after how many days we will remember. And so for that, they have conducted Jost has conducted one small experiment, like they will give a 26 random words uh, to the subjects who are studying with him. And later on, what he will do, after around 8 this timeline is in days. So after around 21 days, he again introduces new list to the subjects. And and he effect affects is their brain with the amnesia electroshocks, it will affect. And after that, how the learning curve is there. So this people, it has been seen that actually the old memories, old list items, they were able to remember more, and the new list items, they were able to remember less. And in apart from time, there has been also how this interference will also impact forgetting. Like for example, I will tell you something, and before you consolidate that new information, I will again uh, give you bombard you with another information. So for this, we'll just try to understand this small list. So there are like four list has been for the experimental purpose. We take around 10 to 15 individuals, or actually 10 to 15 individuals, and we have four lists. So in each list you have around 26 words. So list 1 and list 2, we will give list 1 we will give to the subjects. And before they after 30 around 6 seconds, we'll give the second list. And after 6 minutes, we will ask them to recall. So in this experiment, so they are almost remembering the same percentage of words. Whereas in the second model, we'll give the list 3, and we'll give them around instead of 6 seconds, we'll give them around 60 minutes. And after that, we will give them list 4. So in this process, what happens, the list 3, they in the brain, they were able to consolidate and they were able to remember more. So this, from these things, we can conclude that actually, yeah, if you bombard more and more information before consolidation process, the subject actually forgets the previous information very earlier. So from this thing, we got to know time is important for memory consolidation from these two experiments. And apart from that, consolidation also has a, in physiological part, the organization, how, because in brain all the sensory inputs are going to different parts. So how the organization is happening. So this is one process we want to study. And the psychological part is like, if you if you repeat more and more times, actually the consolidation process is also getting affected. And the association means if you give some meaning to it, some meaningful words, instead of giving random words, if you give some meaningful words which forms a story, so that also has impact. So in so I just want to like tell, from 1900s to 1930s, the first 30 years of 19th century, 20th century, they have concentrated more on interference experiments. Like, because in interference paradigm means, uh, before before the sensory input were getting organized, they were trying to disturb, yeah, they were trying to bombard with more information to the subjects. But in 1940s and 19, in this next 30 years, what they have taken, they have considered the two aspects: how long it takes fixation to occur. Fixation means how long it will take in for the neurons to organize the information, and what kind of physiological processes are critical for the organization. So this part, I think you have already heard from Jayashree ma'am, protein synthesis micro. So this area I'm not going to touch. In this area, people have conducted this three class, means there are many classes of experiment. So for our discussion, I will take three. So what happens is, like, once you get the sensory inputs, uh, before consolidation process, they are giving electrical shocks. So for these experiments, they have studied human subjects, rats, monkeys. And I know this next class of experiments is like, you you decrease the concentration of oxygen in local environment of the subjects, and the temperature changes also. So this, how these three factors are affecting the consolidation process, and how long, how much time it is taking, this was monitored. And the main key findings are like, uh, they are getting inconsistent result, like, for it, because it was depending upon kind of study you are doing, and we are not able to get any conclusion studies. So from this, because before we were just fixing to this model, simple fixation model means, you get the information, and it will take some time to organize, and it will stay forever. Once it is get organized, it will stay forever. So this was previously we were thinking. So here, instead of fixation model, now there is one person, Ips. So he has proposed, instead of forming and consolidating, there is actually a transition. Like, there is actually a transition place. Like, for example, in chemical reactions, you all know, from reactants to products, you have a transition state in between. So in the similar notion, you have a, instead of permanent consolidation, you have a temporary consolidation is also there before. So this idea was introduced, and this Brown, he has studied about this short-term memory and process of consolidation. He has conducted many experiments, and he concluded that disturbance is highest immediately after learning and declines rapidly with age. So the experiments we were doing, the to interpret that results, uh, if you give if you give the disturbances, the what to tell, the consolidation is highly affected at the initial step rather than at the later stage, later time. So all that experiments were done, uh, seeing retrograde amnesia, means disturbing the consolidation process, the memory events before. Now in 1970s, they have done about lateral, but here there was so many conflicts. They were telling, whether it is in the consolidation process, whether it is in the retrieval process. We have three stages, right? Processing, organizing, first step, second step is storage, third step is retrieval. So here people were getting confused between first step and third step in the things, because in this case, in this disease, or in these cases, they were not able to find any locus of memory, means not particularly able to find any system where they can disturb experimentally with the shocks, with all this. So in I think this is in 1980s. Forgot. So there they have EES for EES formalized a model. So primarily it told it is there, the information is getting processed in the hippocampus, and with time the information is going to neurocortex. So that wise they were able to relate between these cases, like you will remove hippocampus in certain subjects and see, but if some people were able to remember the remember the information because they were given enough time to go information into neurocortex. This was there, and for this to test, they have actually done an experiment on monkeys for 16 for over 16 weeks, and they have given what they have trained is they have trained the monkeys to discriminate between 100 objects. They have taken two groups of monkeys. One is control monkeys, one another one is the subject monkeys which you want to study. So they have trained over all these things, and what they have done is, like, in the subject monkeys they have removed hippocampal the hippocampus they have removed. So for that they have observed these things, like in 12 to the things which they have learned more than 12 to 16 weeks past, that things they both were able to recall, but both were able to discriminate between different objects. But in the information which they have trained within 8 weeks, the hippocampal were not they were not able to discriminate. So this proves their the consolidation model. So another thing is, between this because of this transition memory, there are two terms got involved: short term memory, long term memory, you all know anyway. So this year the confusion has come whether these things are occurring in series manner or it's going in parallel manner, because this interesting observation they have found, like, uh, if you block the short term in the molecular process, if you block the short term processes with inhibition drugs, the long term memory is not disturbed. And similarly, if you block the molecular inhibition in long term processes, the short term things are not disturbed. So it means they are actually going in parallel. So but main thing is the long term memories, they were not able to find any synaptic mechanisms properly. So this was there, and even in beyond hippocampus, how the memory is getting consolidated, that uh, that is still ongoing research. So another next second topic I am coming, that is the memory representation. So in memory representation, the thing is, like, you have brain, different function, different lobes are there. This we all know. So main thing is where the memory is getting, whether the memory is distributed over entire brain or is it distributed only in main parts like hippocampus, amalgam, this locations are there. So this question has two different type of analysis. One is macro level analysis where memory are represented, and another one is how each memory is represented. Means, for example, in chips or in memory in your mechanical systems, means computers, you have specific location for each information you are storing. But is it the same thing with living systems as well? So this was the question. So I So here I will focus more on this part. So there are various thoughts supporting the localization theory. Means uh, memory is just localized in certain parts of brain. There were many because for that, in that way they have classified two classes of neurons. One is permeable neurons and the impermeable neurons, where they have hypothesized the impermeable neurons are the vehicles of memory. This is one set of scientists they are thinking. Another set of scientists they are thinking, okay, because in 1824 only he has already established this Florence scientist, as he has established that if you remove certain parts of brain in animals, observed there is no subsequent change. uh, the behavior is not depend upon whether the part is present or not present. So that way it challenges the localized memory theory. And in this way uh, people have now come to the dynamical representation of memory. Means dynamical, in simple words, uh, the memory is distributed in overall brain. Like you have already heard this two key concepts, like presynaptic neuron, postsynaptic neuron. It was also proposed by Ebbs only. And now in more in 21st century we have understood like it is because of uh, biological neural network is the source of memory. So this are the things. So in simple words they have concluded that uh, the memory is actually stored at, as local at global scale, but global at local scale. So very confusing, but this is what they have got to know. So I would like I would like to like we in this chart I would like to just summarize how the things have been grown. Like one thing we know, all the neurotransmitters, all these things are driven by sodium and calcium ions. All these molecules are there, and the channels are there from synapses, and the information is processed in neurons, and the information is also stored in neuronal networks, and from their maps systems, all these higher things also, for long term memory higher things also are involved, this cortex part. So uh, this is just what to tell. For example, there are new if you have in neural networks here, until this point you have clarity, right? Neural different. So after that, in maps means uh, means local, in you will take two three bundle set of neural networks and then form some memory. And systems means this one I not confident about, but this is central nervous system something. Okay. So main references for my all the presentation is these are the two main papers. If anyone wants to explore more, they can read these two papers. And what are the uh, what are the later issues pointed out, and still the things have not been clarity. Though we understand biological neural network is there, but how the things are working, we have molecular details, but at the abstract level, at the psychological process level, because in the physiological process level we are correlating all the things with molecular things, but at the psychological cognition level we are not able to still understand completely how different brain regions forms different forms of memory. So this issue has been reported in this paper. And another mystery, this I only this one I thought of. I don't know whether I didn't explore memory in single organized system. But while going through the journey, I got curious about whether how we are explaining the memory as biological neural networks, but how about single organized system, because they also they also do all the motor functions of the body, whatever they are doing, and they also take the respond to the environment. So how they are doing that? So this this area I will explore more in future. Maybe I will read more, but you can also, if you know anything, you can tell. And okay, so other thing is for if you go with the molecular level molecular theory hypothesis, the main mystery lies is like there has been this two there there has been group of scientists in University of Virginia. They are actually studying previous life memories. So many reported cases has been there. Just to quote, these two books are there. So just to quote this James Lininger, a young boy who had verifiable past memories of being a pilot in World War II. So how these things are there? If we explain everything through physiological process, how we can explain this type of memory? So I hope you can explore more in this area also. And another thing is like I will just give a quick overview of this. This is like we have more or there are more or less we have understood there is something is there apart from this physical. At that point we got stuck. We So many people are working from last 30-40 years, but still we don't have clarity. So they are telling like for example Francis Crick and Christopher Coach, he actually thought about, from 1990s he is thinking about consciousness, how to represent consciousness, because self awareness is also a type of memory. Means if you want to classify, it is also a means when because we also know we are when we reflect upon ourselves, we also have some, okay, I I have satisfied this person, or I have given some gift to my, like, the emotional part at the self core. That part we are aware about, that type of memory is also there. So consciousness, before like 1950s, all these things were there in like in philosophical departments. But this person, he has want to try to put in molecular process all these things. So this is one class of scientists. And another class of scientists, they are trying to explain consciousness is actually something different from all these biological processes. So this debate is all this study is all going on. And even this Mark Minson is one of the founder of AI. He's also involved in this understanding mind and consciousness things. So for small information, I would like to tell the all this group of scientists who are still alive, they are trying to gather. It is there in online also. They are doing some 3 days conference. If you want to explore, you can explore more about consciousness. So the title of the summer symposium, the neuroscience need a revolution to understand consciousness. So you can explore this also, because consciousness I haven't touched about it. I think you have heard from other presentations also, but you can still explore more here. And even in Bhaktivedanta Institute also has some publications discussing about consciousness. You can also look at that copies. Thank you for your kind attention.